say yes if you're good with that. So I think it started. So chapter 16 really talks about the basis of inheritance at the molecular level. And here we're dealing with, at the DNA level. Now, the whole chapter kind of starts out with an introduction to all of the scientists that contributed to the knowledge that we know about DNA and DNA replication. So we're going to kind of review all of those discoveries real quick and how some of them were discovered, and then we'll move right into DNA replication. So let's go ahead and start that. Now, the key scientists, okay, so again, the point was to determine what was transmitted the information from one generation to the next. What directed the cell to do its cellular activities? At some point, nobody knew what the genetic material was or the units of inheritance. Now, some made some hypotheses, which were confirmed later with experiments. So Thomas Morgan, his group basically showed that there are genes located on chromosomes. Okay, so these are, they suspected that there were, hereditary, but we didn't know for sure. It was proven later, but these were the hereditary units. What him and his group did was create a genetic map and discover that some genes are linked or transmitted together from one generation to the next. So we'll see some more of that when we get to the genetics section. However, it is important to know that he did kind of not really start this, but he did provide some, hypotheses about this. And this got other scientists thinking about it if they weren't already thinking about it. So Morgan studied fruit flies. And he studied inheritance between fruit flies. Now, the candidates of inheritance started out with being DNA or protein. So nobody knew what it was. It could have been the DNA, it could have been the protein. So there were other scientists that actually set out to determine which one it was. Now, in terms of the DNA, the role of DNA, it was discovered by studying bacteria and how bacteria can change and be transformed into something else. And it was studied by using things that basically utilizing them in the lab. They're called bacteriophages. They're actually viruses that infect bacteria. So they were able to remember. I don't know if you know much about viruses, but some viruses, they're going to basically get into the cell, and they can either inject their genome into the cell, or they get into the cell themselves and can put parts of their genome into the host genome. Okay, so it incorporates their genome into the DNA itself. But the bacteriophages are specifically viruses that infect bacteria. Now, Frederick Griffith and his group, they discovered that that bacterial cells can be transformed from something that is not pathogenic to something that is pathogenic. And Griffith and his group suspected that it was the DNA that was doing it, but it wasn't sure. And again, still, it could be the DNA, it could be the protein. We don't know. But he investigated 2 strains of bacteria, and these 2 strains of the same species of bacteria were labeled the S and R strain. So S stood for smooth, and this meant the outer covering of it, referring to really a capsule. And the other one was rough. Now, the smooth one was pathogenic. Okay, so what that means is that it causes disease, and it can be deadly. Now, again, the agent or the molecule that was suspected to transform these bacteria from the rough or the non-pathogenic to the pathogenic was not quite identified. However, it was suspected that it was DNA. So here, looking at his set of experiments, we can see that he injected mice with the different strains of bacteria, and these represent all of the controls. So what happens here is the S cells, remember those are the pathogenic ones. This is basically a positive control where you can see that the cell, of course, is expected to die because this is a pathogenic strain. And then this would be the negative control where the living, the R cells, which are the non-pathogenic strain, is injected into the mouse, and the mouse is expected to survive because it's not pathogenic. Well, he killed the S cells, okay, and this is an example of a control as well. It was already known that heat killing, or basically applying very much heat so that it kind of breaks down the cells, and then injecting the mice, that they would still survive that. Okay, so the heat killed S cells, which are the pathogenic form, were basically killed so they cannot cause disease. Okay, so these are the three controls of the experiment. Then what did he do? He combined the heat killed cells. Okay, so you can see this heat killed cell, and this is the pathogenic cell, however, it's heat killed. And he combined the R cells, which are the rough ones, which are non-pathogenic. So he combined those and then injected the mouse, and the mouse died. And then basically he was able to extract living S cells, which are the pathogenic form. Okay, so combining the heat killed S cells with the R cells, which are non-pathogenic, basically the R cells were transformed into S type, which caused death, right? So that caused disease. So in some way, these R cells were converted to something that was non-pathogenic to the form that was pathogenic in vivo, inside the mouse. Now, again, at this point, it wasn't determined whether it was DNA or protein that actually transformed it, but it's still the suspect, right? So either one of those could have been it at this point. Then we have Avery, McCarty, and McLeod. Okay, so they announced that it was DNA, and this is based on some evidence that really isn't described much in your textbook, but they described evidence that only DNA worked in transforming the bacteria from the harmless R strain to the pathogenic S strain. But it wasn't really until the Hershey-Chase experiment. Okay, so this is really a key experiment. If you ever take another molecular biology course in the future, this is something that you should know, right? So this experiment confirmed that it was DNA. Okay, so we have Hershey and Chase that did this key set of experiments. This was done in 1952. So it was in the 50s that we discovered that it was DNA that was the hereditary material. And in this case, transforming the bacteria from the harmless strain to the harmful strain. So they provided more evidence, and this is the group that actually used the bacteriophages. So the T2 phage, and the question was, is it DNA or is it the protein that is actually being transmitted between the cells? So what did they do? I'm going to describe this experiment. So we have two Erlenmeyer flasks, and they're colored differently. Okay, so we have one that's pink and one that's blue. Now, the pink one is where sulfur was basically they incorporated into the medium a radioactive sulfur. Okay, so they can track this radioactive material. Remember, we talked about isotopes. Okay, so radioactive isotopes that can be used in studies, and this is an example of one being used. And then in batch 2, so these are separated, and then radioactive phosphorus. So sulfur is found in proteins. Okay, so if you remember the cysteine, which has that sulfahydryl group, that's found in proteins. It's not found in DNA. But phosphorus is found in DNA. Okay, so the radioactive phosphorus would label the DNA so that they can track the DNA in these cells, and the radioactive sulfur would label the proteins. Okay, so these are two separate things being simultaneously created so they can track where the proteins are going and where the DNA is going. So we have these phages, and inside here we have bacteria that are being infected with phages. And again, the phages are going to inject their genome into the bacteria. And they're basically going to transform that bacteria. And in this case, these phages are labeled with radioactive protein. These phages are labeled with radioactive DNA or radioactive phosphorus. So here the DNA is going to be labeled, here the protein is being labeled. So if we look at the next step, the cells are going to be, here it just shows a blender, they're basically going to break open the cells. So here, if it was protein, then they would expect that the phage DNA or phage, the protein, if the protein was being transmitted from one generation to the next, then these cells would have radioactive material in them. And then, but they're going to take the radioactive material out. And then here, these cells would have them if it was DNA. So they're going to open up the cells. So now when they open up the cells, there's a bunch of membrane material and proteins, and the cytoplasm is going to be, and it's going to be incorporated into the liquid medium. And so basically the membrane and proteins and the cytoplasm are going to be separated from each other. And how do they separate it? They centrifuge them. So what a centrifuge is, it's an instrument where you put the tubes inside and then you turn it on and it spins really, really fast. So it spins them at an angle so that the heavy things are going to fall towards the bottom, okay? And the heavy things are going to be the pellet, okay? This is all the material that's not I guess, dissolved in the liquid. Okay, so it's all the like membrane material. And again, both of them are going to have a pellet, but then they looked at where the radioactivity was occurring. So here we have the radioactivity phage is going to be in the protein, and here the radioactivity was found in the DNA. Okay, so they can actually track where the radioactivity was. So here, if it was injected into the cells, as would have been done here, then you would see all the proteins showing the radioactivity, but they didn't. They saw that the DNA, these ones where they could see the radioactivity was going to be inside this tube right here, where the DNA was actually transmitted. So this provided very strong evidence for DNA being the transforming material, or the material that's transmitted, in this case, from a bacteriophage to the bacterium during infection. Now, Erwin Chargaff was a scientist that didn't really contribute to, DNA being the transforming material. However, he did contribute to some important information about DNA. And this is important information that you should remember from chapter 5. And it's related to the bases and which ones match up with each other, hydrogen bond with each other. So he reported that between species, the composition or the percent of each base differs. Okay, so there's differences between species in terms of, let's say, the percent of adenine versus the percent of guanines. So the percent of each base was measured in various species, and that's what he discovered. Now, this evidence right here basically shows that there is diversity in the DNA, okay? And this does lead to a conclusion that DNA is more likely the source of the genetic material that's transmitted from one generation to the next, if you can see differences between species. If it was the same between species, then they wouldn't suspect that this was the material that actually stores the information that directs the cell. He also basically came about with one of these, he came up with one of two rules. Okay, so the first one, of course, is that the composition of DNA is going to vary between species. And you can see this in the table that's in your textbook. So you can see E. coli, wheat, sea urchin, salmon, human. So we have all these different, in ox, sorry, I skipped that one. And we see that the percent adenine is different between each one. So we've got 32, almost 33% in the sea urchin, however, only 24 to 25% E. coli. And then, of course, if you look at each of the other columns, there are differences in guanine, cytosine, and thymine. Now, his second finding was that the number of adenines about equaled the number of thymines, and the likewise occurred with the guanine and cytosine. So he found that within the genomes of these species, that the percent adenine was about equal to the percent thymine, and the percent guanine was about equal to the percent cytosine. And if we look back at this table again, we can see, I just kind of highlighted guanine and cytosine because they were closer together. So we can see that in E. coli, guanine is about 26%, and cytosine is about, again, if you rounded it up, it would be about 26%. Same thing if you go down here to wheat, about 22%, about 22%. So they're never exact because there can be basically pairings that are not following that rule, but for the most part, they are exact. There can be mutations, is what I'm trying to say here. And so this is his second rule. Okay, so he discovered that. So I have a question, and this might be kind of a sample question that you might actually see in on a test. You know, so here it says, what is the percent thymine in wheat DNA? So if you look at this table right here, you're given, the percent of each base for sea urchin, salmon, and some of it for wheat. Now, I have a poll thing here. Hopefully you'll be able to see the polling and the question. So if you can, try to calculate that and then just select the answer that you come up with. Basically, you want to find the number that would most likely fit in this spot right here. All right, we'll give you another 30 seconds or so, a little bit more. We'll wait till about 2 minutes. So again, you want to find the percent of thymine in wheat DNA. It's basically this spot that's missing right here. You just want to find the number that should go there. All right. So I'm going to go ahead and show the results here. So it looks like we have a small percentage of students choosing A and a larger percent of students choosing C. So if we look at A and C and then we look at the table. Now remember, Chargaff found that the percent guanine equals about the percent cytosine. So here we can see 21, almost 22%, again, almost around 22% for cytosine. So that kind of equals out. So that means that the percent adenine should also equal the percent thymine. And they should total 100% because that is basically the percent of the entire genome. So for this one, since the percent adenine should about equal the percent thymine, so your best answer would be C, in fact. So it looks like the majority of you chose the correct answer. So whoever chose A, I hope that this helped and you understand that why the answer is C over A. So I'm going to stop that. Now, let's go ahead and look at another one. I'm going to actually have you calculate this. So that should be that. So now I have a question here. It says, suppose a double-stranded DNA molecule was shown to have 30% guanine. What would be the expected percentage of adenine bases in that molecule? Okay, so if you know the percent of guanine, how would you calculate the percent adenine? Okay, so I'm going to have you do that. And there's no way to enter, anonymous things inside the polling software. So if you are confident, or even if you're not, just throw up whatever answer you come up with, right, into the chat, or you can say it. Just if you're going to say it, make sure you unmute yourself so that you can do that. So we'll give you a couple minutes to try to work this one out. You might need a piece of paper to do it. So you can go ahead and throw your answer in the chat once you come up with something. So we've got one answer. Let's see what everyone else come. Oh, we got two of the same answer. Three of the same answer. We'll wait a little bit longer. I'm going to go ahead and kind of explain and work it out for those of you who really are not sure where to start with this one. So I'm going to go ahead and do this. Let's see if I can move this in. So we know that the guanine equals 30%. Okay, so if guanine equals 30%, okay, and guanine and cytosine percent should be about equal, we can say that cytosine also equals 30%, right? Okay, so that gives us a total of 60%. Now, again, all of a genome would equal 100%. So if you basically subtracted 100 or 60%, right, from 100%, that leaves you 40%. Okay, so 40% is left. Now we know that adenine should equal thymine, right, based on Chargaff's rules. So if adenine equals thymine, then basically these two should be equal. So if you split 40% in half, right? So then adenine would be 20% And then of course, thymine would make it 20% as well. Okay, so that's kind of the long way of doing it, but it's the way of kind of thinking it out. And, in case you didn't know where to start. So does anyone have any questions regarding that right there? Okay, so you might have a question that's very similar, okay, at some points, either on the unit exam or even on the final exam. And you probably get some practice in MyLab Mastering as well. So again, that's going to be the way to work it out. So if I instead of adenine, if I asked for cytosine, then you would stop right here. So cytosine would equal 30%, right? So you only need one of them to be able to determine the answer, to you basically determine what the percent of all the other ones are. All right. So are there any questions? If there are, feel free to raise your hand or throw it in the chat. Give you a couple minutes to think about it. And so 20%. All right, so I'm thinking everyone kind of understands how to calculate that and understands Chargas rules. And hopefully, if you do come across a question like that you'll be able to answer it correctly. Okay, so again, at this point, the basis for these rules were really not understood until, again, Chargas rules, and until the structure of DNA was determined, right? Until the discovery of the fact that DNA is in the double helical form. So this was kind of talked about in chapter 5, but we're going to talk about how it was actually discovered. Now, it's important to, before we go to the actual discovery of the overall structure, to mention Wilkins and Franklin. It was really Rosalind Franklin's work in X-ray crystallography, which allowed the other groups, basically it was a combination of two separate groups, which allowed the structure of DNA to actually be determined. So at this point, DNA is now accepted as a genetic material, leading up to all of, basically showing all the experiments that are leading up to this. It provided very strong evidence for DNA being the genetic material. So now the big question is, what is the structure of the DNA, the genetic material? So this is what these scientists kind of set out to determine. So again, how is the structure? How does it account for this ability to transmit information and direct the cell? So Franklin, she was an X-ray crystallographer, and she had Wilkins, which was her partner, and she generated this photograph right here. And this photograph, now I'm not an X-ray crystallographer, but this photograph right here basically signified that DNA was in a helical format. Now, at the same time, Watson and Crick were trying to determine the structure of DNA. And what they thought initially was that the pairings of the DNA were basically purine to purine. So remember, purines are the bases which have the two rings, and the pyrimidines are the bases that have the single ring, the nitrogenous ring. Now, they thought that the purines connected together and the pyrimidines connected together. However, this overall structure did not match up with the X-ray data from Franklin's data. Okay, so they originally hypothesized this, but then with the help of the X-ray data, they were able to determine that it was a purine that connected or hydrogen bond with the pyrimidine. And then on top of that, adding in Chargaff's rules with the adenine and thymine, okay, this showed that adenine binds to thymine by hydrogen bonds. Adenine is a purine, thymine is a pyrimidine. It kind of equals up to the combination of all these data from Rosalind Franklin and Watson and Crick. So they adjust their model. And of course, this is based on the image from Rosalind Franklin, right? So from this image, Watson and Crick were able to determine, well, using these images, basically stating that the DNA is going to be in a helical format. They were able to determine the width of the helix and the spacing of the nitrogenous bases, right? So the spacing between each other in the same chain, and also the spacing between the basically between in the separate chains, the hydrogen bonding spacing. So they were actually able to determine the structure of DNA from this X-ray data. And again, the pattern is what suggested that it was a double helical form. And then, of course, Chargaff's rules. It explained Chargaff's rules as well. So all of it just kind of came together at some point. And so Watson and Crick, they introduced the double helix model for DNA. And we know now that that's the accepted model with tons more evidence, which suggests it. So many of these scientists earned a Nobel Prize in one area or another for these discoveries. Now, again, this kind of summarizes Watson and Crick, what they determined with their model. And they determined that the purine binds with the pyrimidine. And in this case, an adenine is a purine, so is a guanine. And they're going to bind hydrogen bond with the thymine and cytosine in DNA, respectively. And you can see the hydrogen bonds, which are formed. So between G and C, or guanine and cytosine, there's 3 hydrogen bonds. And between adenine and cytosine, there are 2 hydrogen bonds. So the bonds between guanine and cytosine are much stronger than the bonds between adenine and thymine. And again, this explains Chargaff's rules and what he found in terms of the composition, the relative composition of adenine versus thymine and guanine versus cytosine in the different species. So again, all of it just came together at some point, and now we have lots of different evidence from different types of studies which show that DNA, of course, is the genetic material, and now we have the structure of DNA. Now the next question is, how does it replicate? Right. So again, cells are going to reproduce, and in order to reproduce, each cell must have the same amount of DNA in it. So at some point, DNA is replicated. So there are different models in which that occurs. So this kind of shows here the overall structure of DNA. So you can see the space filling model of the helix, the double helix form. And then you can see at the nitrogenous base level, you can see the hydrogen bonding between the nitrogenous bases. And of course, just a Leuven type model. Now, here's just to reiterate before we get started on DNA replication. Here it says who conducted the X-ray diffraction studies that were key to the discovery of the structure of DNA. So I can go ahead and relaunch this polling here so that you'll have something to type into at least. Select one of those. So this just kind of gives an example of a question that might be generated about the scientist, right? So it's important to know the scientist's name and what did they kind of contribute to the discovery about DNA in whatever form, and how did they do that? So what type of study? Did it involve radioactive atoms? Or did it involve X-ray diffraction studies? Did it involve bacteriophages? So those are things you want to kind of focus on. I would recommend creating a table of the scientists, right? So if you have the list of all the scientists or groups of scientists, if there is a group, those are working together, and then describe their study, and then what was their ultimate what did they find? So how did they contribute to this area of science? So we haven't talked about Meselson and Stahl yet, but we will. So C should not be your selected answer in this case. So we'll give you another 30 seconds. All right, I'm going to end the polling and we'll share the results. So we've got a couple, A and C. B is the correct answer. It's Franklin, Rosalind Franklin, who performed, she was an X-ray crystallographer, and basically they create these crystals of molecules, and then they perform X-ray diffraction, and they get these images, and it tells you information based on let's say angles and overall three-dimensional structure from these images. So it was Rosalind Franklin that actually kind of contributed in terms of the structure of DNA by X-ray diffraction. Yeah. All right. So again, like I said, we want to find out how the DNA is actually replicated now. Okay, so how is it replicated? There were 3 models that were proposed. So remember, we know now that DNA is the transforming material or the genetic material, and we're trying to find out how it's copied, right? So, and remember, we know that it's a double helix, so there's 2 strands and it's in a helix format. So this was one model. It's called the conservative model, where you have a parent molecule and then the entire parent molecule is copied. Okay, so after the first replication, you would still have the entire parent molecule, and then you would just have a copy of that molecule. So you'd have a copy of 2 strands. This would be like the daughter molecule. And then after the second replication, the daughter one would be replicated. So then there would be 2 copies of that one. And then the parent would be replicated again. And there would be 1 copy of the parent and 1 copy of the daughter. So over time, you would see more copies of the replicated forms versus the parent form. So basically, this is conservative because it maintains the parent molecule. Okay, so this would be the parent molecule, which was just, started the replication. And then, of course, it's exponentially replicated after that. The next model was the semi-conservative model. So the semi-conservative model shows that we have the parent molecule or the parent cell after replication. Each strand is separated and then each strand is copied and create a new type of strand, which would be like a hybrid of the first one. So it would contain 1 strand of the parent strand and then 1 of the daughter strand. And then each of those would be replicated. So over time, you would see a slower increase in the newer strand, but you would maintain the hybrid strand. Now, over time, you're going to see a lot more, however, slower increase in the rate of the strands or the molecules that have the, a daughter type strand, which was just considered a copy. Then we have the dispersive model, which basically suggests that you can see that the parent molecule right here, it just kind of breaks up into parts and then they reconnect. So you have different segments in each of the molecules after that. So then here you would expect all of the ending molecules after several rounds of replication to be the same, right? So they would have a combination of different sections. The parent and the copies. My screen share is paused. Can everyone see the screen? Let me just make sure. It says it's paused. I might have selected something. Resume share. Oops. All right, what's going on? There we go. Everyone can see the screen with the different models of DNA replication, right? Okay, good. Got a thumbs up. Excellent. I don't know what's going on with that. Okay. So Messels and Install are the ones that were in that group right there, although we didn't talk about them, but they identified that it's the semi-conservative model of replication that is actually the case. So we're going to talk about their experiment. So here we can see we have two Erlenmeyer flasks, and here we have isotopes, okay, except different things are labeled, right? So it's nitrogen, because we have nitrogen in the DNA. We already know that DNA is the transforming material or the genetic material. So we want to label only the DNA. So we're not going to use sulfur. We're using only nitrogen in this case, however, different isotopes of nitrogen. So we have the heavy isotopes. We have bacteria that's cultured in a medium. Okay, so this is basically a liquid medium which provides food for the bacteria to replicate. And it's cultured with nitrogen in it that's labeled as the heavy isotope, which has more, remember, neutrons. Okay, so it's going to be a heavier form. And the weight of it is going to matter later, and I'll explain why. So then we have bacteria, this bacteria from the heavy isotope. So this is the initial round of replication. Okay, so the parent molecules are going to have the heavy isotope as opposed to the lighter isotope after additional successive rounds of replication. So they're going to be transferred to a medium that doesn't have the heavy isotope in it. It has the light isotope. Okay, so this is going to be nitrogen 14. So there's a difference between the two at this point. So they can actually measure, and that kind of shows the three models right there. You have the darker blue strands, which are the parents, right? And that would contain the heavy isotope. And then the lighter strands would have the lighter blue strands would have the lighter isotope. So if we look at the next couple steps, we have basically after rounds of replication, the bacteria are going to be centrifuged. And remember, I talked about centrifugation. This is basically putting the bacteria, which is in the medium, inside a test tube and then spinning it really fast, right? So like your washing machine spins around and it wrings out the water. In this case, they're going to be at an angle so that it spins really fast. And the heavier things are going to be closer to the bottom of the tube versus the lighter things, which will be closer to the top of the tube. So then we can see that after these rounds, they're going to be sent. So you can see this is centrifuge after the first replication. This will show basically the base point where all of them would be heavy. And then after the second rounds of replication, you're going to see whichever, it's basically the second round of replication that's going to tell you whether it was the conservative model, the semi-conservative model, comparing between the two, and the dispersive model. All right, so this is basically the results that you would expect, right? So you have the less dense ones, or the lighter isotopes or molecules, are going to be closer to the top of the tube, and the more dense or the heavier ones are going to be closer to the bottom of the tube. So looking at this, after the first round of replication, after you look at the results, right, this is what was found. Okay, so you had a band which showed radioactive energy somewhere in the middle of the tube. Okay, so you can see that this was a mixture of the parent molecule, and it was like a hybrid mixture. So the conservative model was basically excluded at this point because the first round of replication suggested it was either the semi-conservative or the dispersive model. Then after looking at the second rounds of replication, of course, this is going to be excluded as well. But after looking at that, for the dispersive model, you would only see one band, right, closer to the top, because it would be more dispersive. So you would see more of the lighter isotope within the strands versus the darker isotope. But that wasn't the case. Okay, so what the results indicated that it was the semi-conservative model by seeing that there were some strands that were completely copies, and there were still copies that were still utilizing the parent strand as a template to create, to copy the daughter strands. So you saw a really light band, or light in terms of weight, close to the top, and then one in the middle, which was maintained. Right. So this is how the semi-conservative model replication was identified. So they did use, it was Michelson and Stahl, and this is their set of experiments. They did use radioactive isotopes of nitrogen. Okay, so comparing the heavy nitrogen versus the light nitrogen. So now this is basically the semi-conservative model. This is a summary of it. So we have a parent molecule. And remember, this just kind of represents the two strands, and this is a helical type format, okay, even though it doesn't show it, it looks more like a ladder here, but you can see that the A's and G's are matched up with the C's and G's are also matched up. Well, what happens is during replication, these strands are separated. Okay, so the hydrogen bonds are broken between the nitrogenous bases so that there are two separate strands, and then each strand is actually used as a template to copy, right? So basically, this strand right here is used as a template to start adding nucleotides to create a copy, or basically a complementary copy, right? So because thymine binds to adenine, so thymine would go to the first spot in the copy, and then guanine would go in the second, and so on. So basically, this lighter strand right here is a copy of the strand that it used to be connected to. Okay, so this kind of shows the summary of the semi-conservative model. So this shows kind of how it's done. There's an enzyme. It's called DNA polymerase. Okay, so it polymerizes or synthesizes DNA, and it's called DNA polymerase, and it ends in ASE, so it's an enzyme. Okay, so this is the enzyme that actually catalyzes this chemical reaction. So here we can see we have the template strand, and then we have the new strand that's being generated. And basically DNA polymerase is going to take each nucleotide, and it's going to be recognized by its ability to bond, hydrogen bond, with the base that's next in the parent strand. Okay, so again, the parent strand is going to be called the template strand at this point. And then the new strand is like the daughter strand, which is going to be a complementary copy of the parent strand. And what happens is we have a nucleoside triphosphate. Okay, so what's going to happen is DNA polymerase removes two of the phosphates, and this is called pyrophosphate. And then those are going to be released as individual inorganic phosphates. And one phosphate will be maintained, and that'll make up the phosphate backbone of DNA, and it'll be attached. So then the next one would be attached. So the next one that would go on would be a G, or a guanine. Okay, so it would just keep going until it reached the end of the strand. So before we get into the details of DNA replication, I'm going to discuss the differences between bacterial cells and eukaryotic cells, or prokaryotes and eukaryotes, in terms of replication. Now, remember, prokaryotic cells have circular pieces of DNA, and they usually have one molecule of DNA in their cells, and it's circular. And in order for DNA replication to begin, what needs to form is a replication bubble. And what this is basically the separation of the strands. So we have the parent strands, which are dark here. Those need to separate, okay, in order to use them as a copy or as a template to make a copy. So there's an origin where they separate in bacteria. There's a single origin. And this is called the origin of replication. Okay, so there's a single origin of replication in bacteria. And remember, it's circular. So then we have the replication bubble that forms once the parent strands separate. Once the parent strands separate, again, there's going to be a bunch of proteins. We're going to talk about these proteins and other enzymes that participate in this process. But they're going to start to separate, and replication occurs in opposite directions with respect to the entire molecule. Okay, so that's important right here. It occurs basically in a circle. So it's going to go, this replication is going to go from this way, copying both strands on this side, copying both strands on this side until they meet, right? So they're meeting right here, and then it's going to create these two molecules. And these are the daughter molecules, which are going to be a combination of the parent strand and the template strand, or the template strand, which is the parent strand, and the copy of it, or the complementary strand. Now, this shows a transmission electron micrograph of this happening right here. So you can see the replication bubble. And what the arrows are pointing to is called the replication fork. So the replication fork is really where the direction where it's starting to separate. Okay, so DNA replication is going to occur all the way around this molecule until it meets the one that goes in this direction. In eukaryotic cells, remember, they only have linear chromosomes and there are ends to them. Okay, it's not a circle, right? So they're linear and there's ends to them, and they're a lot larger than the bacterial cells. Well, in eukaryotic cells, there are multiple origins of replication. Okay, so we have multiple origins of replication. You can see them right here. So that means multiple replication bubbles are going to form. Okay, so we have 3 right here, corresponding to each of the 3 origins of replication. And then, of course, replication is going to occur in the same manner as it does in bacterial cells, except remember, this is creating a linear chromosome. Now, we're going to revisit this later, but at the ends of some of these strands, they're not going to be able to be completed. Okay, so in eukaryotic cells, after every successive replication, some of the chromosomes start to lose DNA. Okay, so they start to lose a small section of DNA. And we'll explain how that happens as we go through this. Okay, so I just want to mention that now, and we're going to see why later. Okay, so all these arrows are pointing to the multiple replication forks that are on either side of each replication bubble. Okay, so this compares the overall DNA replication between prokaryotes and eukaryotes. Are there any questions? I kind of pause right now and ask for any questions before we get started. So we're going to start moving into the details of DNA replication. Now there's, like I said, there's a ton of different proteins that participate, and your textbook doesn't even cover all of them. But it is important to know what the function of each protein is. Okay, so all of these proteins and enzymes work together in order to replicate DNA, and also, and we'll talk about DNA repair. There's several different mechanisms to repair damaged DNA or mistakes made during DNA replication. Now, there's some of the proteins overlap in terms of function. So some of them work in DNA replication, and they also work in the DNA repair machinery. So we're going to talk about those. Now let's look at the overall. This shows here a replication bubble, and we can see the origin of replication. Okay, so the origin of replication. Now there's 2 strands, right? And each strand is being created simultaneously. Now, there's what's called the leading strand and the lagging strand. Okay, so the leading strand is this right here. You can see it's pointing to it here. And you can see there is a continuous strand which is being copied. Okay, there's not small fragments. Okay, so the leading strand occurs continuously. Okay, so this strand of DNA right here, the darker parent strand, is being copied continuously, and that's the leading strand. And we can see there's a leading strand on the opposite end diagonally going in the other direction. Now, it seems like it's going in the other direction, but remember, each of these strands, the parent strands, are in opposite directions. So remember the 5' to 3' and the 3' to 5'. Okay, so overall DNA replication occurs from the 5' direction to the 3' direction. And because these strands, these dark blue strands, are in opposite directions, this one occurs in this way, going this way, and this one occurs continuously going this way. Now, this leading strand, again, occurs continuously. And This is opposed to the lagging strand. The lagging strand must be done in fragments, and it's because the DNA polymerase or the enzyme that synthesizes these strands of DNA only work from the 3 to or the 5' to 3' direction. And remember, so if we look at this right here, I'll go ahead and draw this if this might make more sense. Okay, so we've got we've got 5' here, and this will be the 3', and this will be the 5' on this end, and this will be the 3' on this end. Okay, so DNA replication is only going to occur from the 5' to 3' direction. So you can see the leading strand, it's just going in that direction readily. Now, because this is 5' here, that makes this 3'. Okay, and then that would make this the 5' end right here. Okay, so this would make this the 5' end, and then this would be the 3' end. We can't do this way because this would be the 3 to 5 end, the 3 to 5 direction. Okay, so I'm going to erase that. Okay, I'll get back to. Alrighty. I don't want to get out of the eraser. All right, let's go ahead and just clear again. I'm struggling here with this. I'm just going to delete it and then go back to annotate. There we go. So again, this is a 5' to 3'. Okay, this is the 5' to 3', and this is going to only occur in this direction. Okay, so this makes this. This is 5, and then this is 3, and then this is 5 right here. Okay, so these are going to have to occur in this direction. You can see the arrows, right? This one must be done in fragments because overall DNA replication is occurring in this direction between the strands. Okay, so this one can continue continuously, right? It can synthesize continuously. This one's working back this way. So this is basically fragment number 1. This is fragment number 2, and this is fragment number 3. These are called Okazaki fragments. So they're named after the person that kind of discovered them, Okazaki. So I must point out these orange areas, and we'll see another way of looking at this in just a moment. But these little orange or reddish areas are called primers. Okay, what a primer is just a short sequence of RNA. Okay, so this is RNA that kind of gets DNA replication started. So there's an enzyme called primase, and we'll see this in just a moment, but there's an enzyme that actually puts that primer there. And the primer is basically, we have this 5' end and then the 3' end. Okay, this provides a 3' hydroxyl group so that DNA polymerase can start adding nucleotides to synthesize this strand right here. If the primer wasn't put there, it wouldn't be able to add the nucleotides just from nothing. Okay, so the primers are actually required to get DNA replication started. Okay, so that kind of gives you an overview of that. I'm going to go ahead and clear this and we'll move on to the next slide. Now this looks at it a little bit closer. Now we can see the 2 strands, and we have the parent strands, which are in blue. And then they kind of look like a zipper, but each of those prongs on the zipper represent the nitrogenous bases, which are connected together by hydrogen bonds. Now I'm going to talk about some of the proteins that are involved in this. Now, since we were just talking about the primer, okay, it's RNA bases that are added basically complementary to this. So if this right here, let's see here, if this right here was an adenine, right, in DNA, this would be a uracil, right? So this is RNA. Okay, so if this was thymine, this would be an adenine right here. So again, this is RNA. And we'll talk about why it's RNA later, but it's RNA. And the enzyme that puts that RNA primer on is called primase. Okay, so the enzyme primase adds the RNA primer. And the RNA primer is needed to provide that 3' end, which has a hydroxyl group, which allows the addition of each nucleotide. Okay, so the nucleotide will come in and attack that hydroxyl group. And then the nucleotide will be added. So after this RNA primer, it'll be DNA that's added. So let's say this is an adenine, this would be a thymine. Okay, let's say this is a thymine, this would be an adenine. So again, this is DNA that's being added to the end of the RNA. Now, the strands are separated by an enzyme called helicase. So helicase is the enzyme that unwinds the DNA. Unwinds. Okay, so it basically separates the strands. And remember, the strands are twisted in a helical format. So if you imagine taking a rope and splitting all the strands within a rope, it would get tight on one end. It would become very tight and compact on one end. This creates a strain, right? Topoisomerase, there's many different topoisomerases, relieve that strain. So it kind of winds up the DNA. It cuts like gaps in there and then reconnects them so that it relieves the strain that's caused from separating the strands because they're helical in nature. Now, in order to keep the strands separated, the single-stranded binding protein is needed. Okay, so each strand is going to have, many different single-stranded binding proteins attached to them to keep the strands separated. And the reason why you need to keep the strands separated is because they're attracted to one another by hydrogen bonding within the nitrogenous bases. So again, this keeps them separated. So these are a number of different proteins and enzymes that really participate in DNA replication, but there's more. Okay, so we're going to talk about those in just a moment. Okay, so this kind of gets you started. I do have a worksheet, which I'm not sure if I added it yet, but I will add it to the bonus module, which will be extra points to complete. There's a table, and the table has all the listed enzymes and proteins, and you're to describe what their function is. So what's their purpose? And then below the table is basically this image right here where I want you to draw all of this stuff and explain what's happening, but include the other things that are in the table. Okay, so you want to include all the other things that are in the table, and we'll see an image which kind of does that in just a moment. Okay, so this kind of gets the DNA replication started. Now, if we look at this right here, we have the parent DNA. Again, this is the same idea here. We have helicase, remember that's separating strands. And then we have single-stranded binding protein, which are going to keep the strands separated. And then we had primase, which added the RNA primer. Remember, the RNA primer is just RNA nucleotides, which are added in a small fragment. Well, then we have DNA polymerase 3. Okay, so why are we calling that 3? Because there's many other DNA polymerases. Okay, so DNA polymerase 3 does the initial synthesis of the DNA. So then DNA polymerase 3 has this sliding clamp type of mechanism, which just basically slides along the parent strand, adding nucleotides, okay, and basically connecting them together or clamping the strands together. So we have DNA polymerase 3, which is going to continue the extension of DNA onto the end of the RNA primer. Okay, so you can see over time, okay, continuous elongation, and this is the leading strand. Remember, the leading strand is a continuous, it doesn't create fragments. Okay, so we have DNA polymerase 3 that performs the synthesis of the DNA, connecting nucleotides to the end of the primer. Now, I see a question possibly in the chat. Just sorry if I need to restart. Okay, that's okay. So I'm recording this, so if you have to leave and then come back, you won't miss it all. So you can see that we've got the leading strand, and we're talking about synthesis of the leading strand. Again, DNA polymerase 3 is going to take part in the synthesis of the strand, which is adding nucleotides to the end of the primer. So if we move ahead, we're going back, and now we're going to go to the lagging strand. So the leading strand just continues until the end of it is reached. Ooh, let me go back real quick. So if we talk about this right here, let's look at this RNA. Okay. Now, again, it's RNA, and we want this whole strand to be DNA. So imagine this DNA polymerase 3 synthesized DNA all the way to this 5' end of the parent strand, creating the 3' end, which is connected to it. Well, now we have this RNA primer, which is still there. Well, the RNA primer is removed, and then normally DNA is going to be attached. But at the very end of the chromosome, it can't be, because nucleotides cannot be attached to just nothing. Okay, so remember, I was telling you that the ends of the chromosomes start losing DNA after every successive DNA replication. This is why, because at the very end of the leading strand, there is a primer which needs to be removed, but DNA cannot be replaced with it, right? So it can't be replaced with DNA. So again, that short segment is going to be removed during the next successive DNA replication, and then it'll continue on that leading strand through every replication. Now you can imagine this could cause problems over time, and we'll talk about what can happen and what mechanisms are in place to avoid any negative effects from that. All right, so let's move back into the lagging strand. And remember, the lagging strand is done in fragments. So overall DNA replication is occurring in this direction right here. You can see the red arrow down at the bottom. But since these two strands are in opposite directions, the lagging strand must be done in fragments. So basically, there are multiple primers added along the length of the lagging strand as it's being separated at the replication fork. Okay, so there's a primer that's going to start this synthesis of this one. Okay, so we add a primer here. Primase will add the primer, and then DNA polymerase 3 will come in and synthesize DNA until it reaches the primer that was ahead of it. And then we have primer number 2, and then we have fragment number 2. Then we have primer number 3, and then fragment number 3. And then primer number 4 will come up here, okay? And then fragment number 4 will be here. Once this is separated, fragment or primer number 5, and then fragment number 5. So you can kind of get the idea here. It's going to move this way, and then this one will move this way. We have primer number 1 here, primer number 2, fragment number 3, and primer number 3, and then fragment number 4 and primer number 4, fragment number 5, primer number 5. So you can see the lagging strand must be done in fragments. These are called Okazaki fragments. So we're going to look at this in detail. So here we have the template strand, and we have, this was the primer that was added before this one. Okay, now we're talking about this primer. Okay, so this is the first primer, and then this would be the second primer. So this is the enzyme that adds the primers. It's called primase. Okay, and this shows the origin of replication. So this would be the origin of replication and the point where the leading strand going in the opposite direction would start to be synthesized. But behind the leading strand, on that same strand, we had the lagging strand going in the opposite direction. So then if we click to the next segment right here, DNA polymerase 3, okay, is going to synthesize DNA. It's going to attach bases to the end of the RNA primer and create Okazaki fragment number 1. Okay, so now we have Okazaki fragment number 1. This will continue synthesis until it reaches this primer right here. Okay, so we've synthesized Okazaki fragment number 1. Now you can see DNA polymerase 3 synthesizes the leading strand, which is continuous, and it also synthesizes the DNA. Again, it's always adding DNA to the ends of the primers. Okay, so these are fragments. However, it's still DNA polymerase 3. Now, DNA polymerase 3 is going to detach once it reaches that RNA primer. Okay, so this RNA primer is basically either the fragment ahead of it, or it could be whatever synthesis was starting on the leading strand going in the opposite direction. All right, so Okazaki fragment number 1 has been synthesized. Now, if we look at this, we're going to talk about Okazaki fragment number 2. So we have Okazaki fragment number 1 with its starting primer. Well, RNA primase is going to add a primer just behind Okazaki fragment number 1. Okay. And then DNA polymerase 3. Okay, so it's not labeled here, but this is DNA polymerase 3. It's going to add nucleotides to the end of Okazaki fragment number or primer number 2, creating Okazaki fragment number 2. Okay, so this is synthesizing and it's going to stop when it reaches this primer, and then it's going to detach. And then it'll be ready to create Okazaki fragment number 3 behind that one. Now, as you can see, we've got another enzyme here, right? We got this yellow thing right here, but it's not labeled. If we go to this next thing, we have a label for that. Okay, that's DNA polymerase 1. Okay, so DNA polymerase 3 created the Okazaki fragments. DNA polymerase 1 removes the RNA primers and then replaces it with DNA. Okay, so DNA polymerase 1 removes the RNA primer bases and then replaces those bases with DNA. It's DNA polymerase 3 that synthesizes the DNA in the leading strand, which is continuous, and synthesizes the Okazaki fragments. Okay, so DNA polymerase 3 is simply going to add bases to the ends of each of the RNA primers. DNA polymerase 1 basically removes the RNA primers. Okay, so it takes those bases away and then adds DNA bases in its place. Now, if we look at this a little bit further, this process requires another step. So DNA polymerase 1 can synthesize or add bases in place of the RNA. But when it reaches the end of the RNA, it cannot connect those two bases right there at the end. So that would be the new base that's in place of RNA base and the old base, which was part of the Okazaki fragment. So that creates a gap, and you can see the gap here. Okay, so those two bases are not connected on the same strand. Well, the enzyme DNA ligase, so to ligate means to connect, okay, or create a bond. So DNA ligase forms the bond between those two bases. So now we have a whole nother enzyme, which we haven't even talked about yet. DNA ligase. Okay, so once DNA polymerase replaces all the RNA bases with DNA, there's a gap. DNA ligase fills in the gap. Okay, what the gap is basically these two bases are not bound or connected. Okay, so DNA ligase connects them. Okay, so we have that other enzyme now. So once this occurs along the length of the entire lagging strand, then the lagging strand is complete. Okay, so once every Okazaki fragment is connected, it's complete. And now at the end of the strand, we have another piece of RNA, which is going to be removed and replaced, and it can't be replaced with DNA because there's no base behind that to actually attach a nucleotide. So this is another strand that's going to start losing DNA over successive replications. Okay, so we're going to revisit that in just a moment, but let's look at the overall process here. So we can see this shows the leading strand. It's going to be done continuously. Okay, we have DNA polymerase 3 in here. We have the replication fork. Beyond the replication fork, topoisomerase is going to be creating gaps and relieving the strain that's caused from separating the strands, okay, because they're helical, they're twisted, right? So if you separate the strands that are twisted, it's going to create strain up ahead. Now, helicase is the enzyme that separates the strands. And then we have DNA binding protein, which are these gray circles right here, which, or single-stranded DNA binding protein. It binds to the single strands and keeps them separated. we can see that the lagging strand is being synthesized. So up here, primer number 1 or Okazaki fragment number 1 is complete. The primer is already removed. DNA polymerase 1 already replaced that with DNA. And now ligase is coming in to connect that gap right there. Here, DNA polymerase 1 is in the process of removing the RNA primer and replacing it with DNA bases. Once that's done, DNA ligase will come over here and connect the gap. Back here, we have Okazaki fragment. So this was connecting Okazaki fragment number 3 to 2. And then we have right here, DNA polymerase 3 is creating still Okazaki fragment number 4. Once DNA polymerase 1 is done replacing the RNA here with DNA, it's going to go back here and replace this RNA with DNA. And then ligase will connect the gaps as it goes along. So here we can see primase is adding the next primer, right? And this is going to create Okazaki fragment number 5. This is going to separate, then a primer will be added to create Okazaki fragment number 6. And it'll keep going until it reaches the end. Okay, so this kind of shows a summary of that whole process, at least going in this direction. So we can see this is kind of like a blow up of, or basically an overview of the whole thing. And this is a blow up of this replication fork right here. Well, there's another replication fork on the other side, which is occurring simultaneously. Remember, this is the replication bubble, and this is at one origin of replication. Remember, eukaryotes have multiple origins of replication. Now, I know this is a lot of information. There are some videos in My Lab of Mastering, which will be helpful. Okay, so it just kind of shows an animation of the process and kind of describes that process. Those are really good to watch as well. I would recommend drawing out the process, right? So you might want to draw these stages out, right? For each, the leading strand and the lagging strand, right? So it might be a good idea to actually do that. Adding each protein in or enzyme, and then describing next to each protein and enzyme what it's actually doing during the process. Okay, so this kind of shows that overall bubble. We basically talked about what's happening on this side right here. Well, the same thing is happening on this side. And you can see the overall direction of DNA replication is going in opposite directions because remember, DNA polymerase can only add nucleotides from the 5' end to the 3' end. So in the 5 to 3 direction. Okay, so there's a quick question. Hopefully you guys are still not too overwhelmed with this information. Here it says, imagine a bacterial replication fork. Synthesis of which new strand would be affected by mutations in the enzyme primase. So here you want to think about the function of primase, okay? And what would be affected if that enzyme was mutated? All right, so I have a poll here for you. So you want to think about the function of primase and what would be affected, or what part of protein or DNA synthesis would be affected if the primase was mutated so it's not functional anymore. So here you want to think about the function of primase. And I know you just learned about it, but again, these are types of questions which kind of get you thinking about it. And again, it's not asking you what's the function of primase? It's asking you another question where you have to think about the function of primase in order to answer that question. So I'll give you a couple minutes to go ahead and try to think about it and see what your answer is. We'll do another 30 seconds. All right, I'm going to end the polling and we'll go ahead and share the results. So we're all across the board here. And everyone picked pretty good choices, right? So it's completely understandable. But C is the correct answer. And I'm going to go ahead and explain why. Now, it's good that nobody picked D because we know that primase can have a function in either the leading or lagging strand or both, right? So it has a function during DNA replication. So it has to be one, your answer has to be A, B, or C. All right, so I'm going to go ahead and stop the sharing right here. So again, it's going to be C. Okay, so it should be C. And let's kind of go back and go back to this picture right here. So remember, primase is the enzyme that adds the primer to get DNA replication started. And you can see primase has a function in the leading strand and the lagging strand. More so in the lagging strand, however, if primase was mutated, it would affect both. Okay, so I hope that makes sense to everyone. So again, here, this is the type of question where it's not like a direct question about the function of primase, but you still have to know the function of primase to be able to apply that knowledge to this given scenario. Okay, so again, it might not be something that's directly, let's say, talked about in the textbook, but this is something that you want to be able to think about and kind of establish and answer what you think would happen based on your current knowledge. And your current knowledge should be that primase has a part or has a role in starting the leading strand and basically continuously throughout the lagging strand. All right, so hopefully that's a good example of one of those kind of critical thinking type questions or scenario type questions. Now let's look at this one. We have imagine a bacterial replication fork, and it says a synthesis of which new strands would be affected by mutations in the enzyme DNA ligase. All right, so think about the function of DNA ligase. Oops. And I will go ahead and reset this, and you can choose the best answer for this question. So remember, DNA ligase is the enzyme that connects the gaps after DNA polymerase 1 replaces the RNA primers with DNA. All right, another minute. Another 30 seconds. All right. Let's end the polling, and I'll go ahead and share the results. So we have a higher percentage of everyone choosing A, and we have some choosing B. So we have the difference between leading strand and the lagging strand, and then a small percentage choosing neither. Now, DNA ligase does have a part in DNA replication. Okay, so D is definitely not the answer. But let's think about what DNA ligase does. DNA ligase connects the nucleotides after the DNA polymerase 1 removes the RNA primers and replaces it with DNA. Okay, now this only occurs on the lagging strand. Okay, so B should be the correct answer here. And I'm going to go back and explain why. So if we go back to the figure here. Okay, so the leading strand is continuously synthesized. Okay, so this one primer is added, and then basically synthesis of DNA continues until it reaches the very end of the chromosome. Now, this one primer will be removed, but DNA cannot be replaced with it because there's nothing to add the nucleotides to. Okay, so the lagging strand has the Okazaki fragments. So basically, the DNA ligase connects the Okazaki fragments after the RNA primer was replaced with DNA by DNA polymerase 1. So it's only going to have a function on the lagging strand. It also has a function in DNA repair as well, but we're only talking about leading and lagging strand during DNA replication. Okay, so hopefully that makes sense. Okay, so in that case, it would be the lagging strand. We're going to talk about proofreading and repairing DNA, briefly about proofreading, but repairing DNA after a break. So let's go ahead and take a break. Let me go ahead and pause this real quick. All right, here we go. We're getting started. So at this point, we've discussed the entire process of DNA replication, replication of the leading strand and the lagging strand, and really the differences between the two, and explained the function of all of those proteins and enzymes that are involved in that process. Now, DNA polymerase, not all DNA polymerases do this, but in general, DNA polymerase can proofread itself. So if it makes a mistake, it can address that mistake right away, but sometimes it doesn't, right? So sometimes a mutation occurs or a mispairing of DNA bases will occur and it needs to be fixed, okay? So this is part of DNA repair or, part of the proofreading process. So again, some DNA polymerases can proofread themselves, but some need other machinery in order to do that. Now, sometimes damage can occur to DNA, and this damage can be like chemical or physical, like X-rays, right? So damaging UV rays. So those can cause changes in the DNA, in some cases adding like functional groups that cause I guess it will eventually cause mispairing of DNA bases. So again, it can cause mutations as a whole. And this needs to be fixed, right? So the cell actually has machinery that allows the fixing of this. Now, not everything is going to get fixed, right? So if there's a mutation that occurs, it could get passed on to the next generation. And if it is passed on from generation to generation and it doesn't affect the viability of the organism, then it'll just be another change in DNA. So I think I might have a question here. Okay, so I will definitely explain the leading and lagging strand again, but I think I'm going to do that at the very end. And with the extra time, hopefully that's okay. You do not need to really know the leading and lagging strand in order to understand the rest of this. That's why I can easily go back to that. So what I will do is describe the whole process again as a whole once I'm done talking about the rest of this. So those of you who really want to listen to it, you can stay, and then hopefully that'll help out. Hopefully that's good. Excellent. So as I was saying, we will go ahead and talk about the proofreading and repairing of DNA. And then I'm going to talk about the ends of the chromosomes in eukaryotes. And remember that end problem where the RNA primer at the very 5' end of the strand, it's going to be removed, but it can't be replaced with DNA. So then, there's going to be successive shortening of the chromosomes over time. We'll talk about the ends of chromosomes and how that can be addressed. All right, so let's talk about proofreading first and repair. So as I mentioned, I already discussed the purpose of proofreading and repair. So there's a couple different repair machineries. So there's what's called mismatch repair. You might see something, if you're reading somewhere, it might be capital MR, or MMR. That's mismatch repair. So this is where you have repair enzymes, which are going to recognize mistakes in the base pairing. And usually the mistakes in the base pairing can be recognized by there like being a bulge in the overall DNA molecule where there shouldn't be a bulge. Okay, and it's due to mispairing. Let's say instead of matching a pyrimidine with a purine was matched with another purine. Okay, so that is one machinery. And then there's the nucleotide excision repair. And it's called NER. Okay, so you might see it abbreviated as NER. So this is where you have repair enzymes, which are going to recognize damaged DNA or mutated, right? And mutated meaning that some of them can be damaged where it could create these thymine dimers, which I think are mentioned in your textbook. This is caused by UV radiation. And then sometimes chemicals can cause changes in DNA, the chemical structure of DNA. For instance, an alkyl group could be a functional group could be added to the DNA, which again is going to cause a bulge in your DNA strand, which will be recognized by these repair enzymes. So again, nucleotide excision repair is going to be a set of enzymes that's going to recognize chemical or physical damage or changes to the DNA. And mismatch repair is really meant to correct mistakes. Now, the overall mechanism of this is the following. So we can see we have the DNA strand and we see a bulge right there. And this actually shows these two nitrogenous bases bonded together, creating this bulge. And this really represents a thymine dimer. But again, you can see a bulge there. So we have an enzyme, which there are specific names to these enzymes. There's enzymes that are going to recognize the bulge and then attract a type of nuclease. So a nuclease is a specific enzyme which breaks up nucleotides. So this is going to be an enzyme that actually removes nucleotides, basically removes this damaged part and sometimes some bases around it. So it removes some bases like around the damaged part and the damaged bases after it's recognized and basically kind of summoned over to this damage. So it's going to remove the bases. And then we have DNA polymerase 1, which is going to replace the damaged bases with the proper bases. So those would be new bases and hopefully matched up properly between this would serve as the template strand. And then, of course, we have the new bases that are being added by DNA polymerase 1. And then after that happens, again, we're going to have a gap there. And remember, just like in DNA replication, the thing that connects the bases that create the gap is called DNA ligase. So DNA ligase has another function, right? And so does DNA polymerase 1. So DNA polymerase 1 has a function in DNA replication, and it has a function in DNA repair. So does ligase. Right, so both of them have functions in DNA replication and DNA repair. So this is the general mechanism, but there are more specific mechanisms and specific proteins that are involved in this, but your book doesn't really get into much of the details. So if you are studying in a more upper level type of molecular biology course, you'll probably be introduced to more proteins and a more intricate detail of this process. All right, so this is a way of fixing mismatches, right? So basically, let's say A and T were not hydrogen bond together. It was like C and T, or C and A or something. And then that can be replaced and also damaged DNA. Now, we're going to revisit the ends of the DNA molecules, right? So on some strands, the entire chromosome can be replicated. Well, on one end, right? So the other end, for instance, in the leading strand where the primer is added, it cannot be replaced by DNA because there's nothing to attach the bases to. Okay, so DNA polymerase needs a 3' hydroxyl group in order to add a nucleotide to it. So this is a limitation to DNA polymerase. It can't just add nucleotides to the 5' end of a piece of molecule or DNA molecule. It can only add to the 3' end. And again, this creates a problem for DNA that's linear, right? It's not a circle, so the gap can't be filled, right? So it's got ends, and some of the ends are going to have RNA, which is eventually going to be removed, and DNA cannot replace it. So the usual replication machinery basically has no way to add on to those 5' ends of the strand, right? So it has no way to complete the 5' ends because the RNA is removed and it can't be replaced with DNA in that direction. So this kind of shows this thought. Okay, so it's not needed to know or understand the leading and lagging strand at this point. Just know that we can see that we have the ends of the parental strands right here. And we can see what happens when we look at a close-up of the end. So we have a close-up of the end here where there's an RNA primer at the end. So here, this is in between the fragments. So this RNA can be replaced by DNA using DNA polymerase 1, and then the gap will be filled with DNA ligase. Well, this RNA primer is going to be removed, but then that leaves a 5' end. Okay. Bases cannot be added to a 5' end. They can only be added to a 3' end. You can't just add one here. You need a 3' end on this strand, which can't be done. Okay, so once the primer is removed, this is shortening of that strand. Okay, so that strand is shortened. And then the next round of replication is going to separate these strands. Okay, and then this strand is going to be synthesized. It's shorter, but then the next strand that's using this as a template is going to be shortened as well. So over time, you're going to have more shortened strands, and then they're going to continue to be shortened over successive replications. And this only occurs in the linear chromosome, on the linear chromosomes. The circular chromosomes are going to meet at some point, and they can fill that gap right there. The linear, they have the ends, right? Circular chromosomes do not have ends, right? So this creates successive shortening of the chromosomes. Now, there is, what can happen here is if you shorten the chromosomes far enough, you're going to start losing genes, right? So we have genes which are needed to produce products which have a function inside the cell. If you start losing the DNA that makes the product, then you're going to lose function inside your cell and the cells can die. Okay, so eukaryotic chromosomes have telomeres at the ends of their chromosomes, which are kind of highlighted in this fluorescent image right here of the chromosomes. The telomeres are highlighted in the yellowish orange color. Okay, now the telomeres, and you can kind of see where the centromere is, it's kind of pinched off right here. It's very tight where the centromeres are, but the telomeres are at the ends, and those are also very compacted. They're very tightly compacted. There's no gene material on the telomeres. What the telomeres are nucleotide sequences, okay? But they're just repeats of the same nucleotide sequence. They do not provide a code to make any proteins or any functions inside the cell. The main function of the telomeres is to add bases so that when the chromosomes are successively shortened during DNA replication, you're not losing gene material, you're only losing some of those repeats. Okay, so it prevents erosion of the genes. So erosion would be subtraction, right? So reducing of genes, it prevents that, or it slows the process down. Now, it's been discovered, I actually had a friend that was studying the length of telomeres and aging. And it's been studied. I'm not sure what the most recent discoveries are. However, I think what I read at some point was it was mixed in terms of the results, but it is suspected that those individuals that live longer are more likely going to have longer telomeres. Okay, so, and I guess part of the hypothesis would be is that if you have longer telomeres, then it takes longer, or you can have more, I guess, successive replications without losing genes. Now, other things can kill people, right? So again, you can imagine that there's so many variables, but the length of telomeres have been associated with aging or lack, longer age before death. Now, also, telomeres have a correlation with cancers as well. Now, there's an enzyme called telomerase. Okay, so telomerase is the enzyme that adds those repeats to the ends of the chromosomes. Okay, so it adds that DNA, those repeated pieces of DNA to the ends of the chromosomes. So that's an enzyme that does that. Well, if you have overall or overactive telomerase, then you could continually be creating basically so the cells can live longer, right? Okay, so if you have longer telomeres, your cells can live longer. Well, that's not good if you have cancer, because you don't want those cells that are cancerous, that have been transformed into cancer cells, to be able to replicate, right? Or reproduce and create tumors. So it's actually been discovered that some cancers have excessive telomerase activity. And this helps the cells be able to survive and keep reproducing without losing gene material and dying eventually. So again, telomerase can be good and it can be bad, right? So it can help you live longer, but however, it could help the cells that can produce tumors live longer as well. So it's kind of like a double-edged sword. Now, I want to reiterate here, and we're going to talk about chromosome rearrangements. So a chromosome is actually chromatin. And remember, chromatin is DNA with proteins associated with it. Okay, so a chromosome is basically a DNA molecule which is associated with some proteins. So we're going to look at the difference between bacterial chromosomes or prokaryotic chromosomes versus eukaryotic chromosomes. So remember, bacterial chromosomes are circular, and they are associated with a small amount of protein, but not as much as eukaryotes. In prokaryotes, the DNA molecules remember, it's circular and it's smaller. It's usually supercoiled, okay? And it's supercoiled and compacted, but not associated with as many proteins. And remember, it's located in what's called the nucleoid region. There's no nucleus in these types of cells, so that it's just in a region where the DNA is, the supercoiled DNA is. Okay, now as opposed to eukaryotic chromosomes are linear. We've already talked about the significance of that in terms of telomeres and loss of DNA material, but they're all double-stranded. It's all the same DNA molecules, but the eukaryotic chromosomes are going to be associated with a lot more protein. And the chromosomes can be very condensed or compacted, or it can be loose, right, in some areas. And the DNA of eukaryotic cells is found in the nucleus, so it's within the nuclear envelope. Now, let's expand upon that. We're going to talk about the chromatin. Remember, chromatin, as I described, is a complex of DNA and protein. And this is typically described as eukaryotic chromosomes because eukaryotic chromosomes are associated with a lot more protein than the prokaryotic chromosomes. And chromatin can be, like I said, can be very compact, or it can be kind of loose in some areas. So this shows a transmission electron micrograph, and you can see this is the DNA double helix. Well, the double helix is going to be associated with proteins called histones. Okay, so they're called histones. And there are several different types of histones. So I'm going to take away this part of the picture. We can see the rest of it. So the DNA is going to wrap around the histones, or a complex of histones, creating nucleosomes. So we can see the double helix right here. And then now we have the double helix that's wrapped around the complex of histones, creating what looks like beads on a string. Those are called nucleosomes. So this is one nucleosome, this is another nucleosome, and this is another one. Then the nucleosomes start to connect or interact with each other. So what is significant about this in terms of charge? Remember, DNA has a negative charge because it has that phosphate backbone. Remember, phosphate is negatively charged. Well, the histones are positively charged. Okay, so negative and positive makes it neutral. So we have the negative DNA wrapping around the positive the positive histones, or complex of histones, which neutralizes the DNA, and it allows these nucleosomes to interact with each other by hydrophobic interactions. So this is what happens when the chromosomes become condensed during mitosis, right? So the DNA is wrapped around these histones, and then the histones can interact with each other, or they could not interact with each other. And it's the histones that are modified. They're modified by adding or taking off a functional group. Okay, so let's look at this a little bit further. We've got the nucleosome, we've got the beads on a string. These beads on a string are going to connect to one with another and create these long fibrils. So here we can see the fiber. So this is a chromosomal fiber. This is a very condensed piece of DNA, and it's condensed because the DNA is interacting with the histones. And the histones are interacting with each other. So it creates, it kind of scaffolds upon itself and folds up, and then creates this condensed chromosome. And we can see that it's starting to scaffold even more, and like a three-dimensional level. And this is what creates the mitotic chromosome. So we can see, remember, this mitotic chromosome here is actually two chromosomes. One of them was copied. They were both condensed, and now they're interacting with each other, right? So those are eventually going to be separated during mitosis, and then they'll become individual chromosomes. So if we go back to, let's say, the process of mitosis, right? So during the process of reproduction of cells, we could say that during prophase, this is what's happening. Okay, so the DNA is starting to compact. Okay, so during prophase, and then later on in prophase, it's creating more compact pieces of chromosomes or chromatin. And then by metaphase, you have this right here. You have the two chromatids tightly compacted and connected together, creating the metaphase chromosome. And it's these metaphase chromosomes that are extracted. So the cell is stopped at this stage right here, so that you can extract the metaphase chromosomes and line them up to create a karyotype. So this shows a karyotype where each of the chromosomes were labeled with different fluorescent molecules. So they were kind of labeled by size, right? And then matched up. And we can look at the chromosome, and you can label certain sections and highlight like mutations that might occur. And then here you can determine that this is from a female, right? So there's 2 X chromosomes. Okay, and during prophase or during interphase, your cell would look like this, right? So this is before the chromosomes or the chromatin was tightly compacted. It's all surrounding the cell, just loose in there. And then once it starts undergoing mitosis, it starts to develop into the metaphase chromosome, which is shown here. They're just not matched up by size like they are to the right here. So that shows the process of the condensing of the chromosomes. Now, chromatin usually is somewhat loosely packed. It's not very tight, but there are regions of the chromosome that are very tight. And you've already seen a couple of them. So we have the centromere, so that's like constricted in the middle there. And then at the telomeres, it's very tightly packed at the telomeres. These are regions where there are no genes. Okay, so there's no genetic material, nothing that's going to be used to make some sort of product. But in general, even the parts where there are genes can be loose or it can be more tightly packed. So most of the chromatin is going to be loosely packed in the nucleus during interphase, but then become tightly packed during mitosis. Now, there are names for these, right? So the loosely packed chromatin is called euchromatin. The tightly packed chromatin is called heterochromatin. So the areas that are always really highly condensed are going to be, like I mentioned, the centromeres and the telomeres. And within the euchromatin are going to be where the genes are. Okay, so the genes are going to be segments or regions of the DNA which actually code for something. Okay, and they're going to be used later to make that thing. Okay, so they need to be accessed readily and be able to basically pull them out loosely, right? So that they need to be loose so that it can be pulled out as needed and the strands can be separated and used to make whatever they're meant to make. And I'm usually talking about a protein, but they may not make a protein. They may make an RNA, like the RNA that make up ribosomes, the ribosomal RNA. And so that kind of leads us, and we could see different segments of the chromosome. We have the centromere here, which is tightly packed. And then this is the pericentromeric region. It's very close to the centromere. And then we have the euchromatin area. The euchromatin is going to be really where most of the genes are located. And then we have the subtelomeric region, which is very close to the telomeres. And then we have the actual telomeres. Here, there will also be euchromatin, the same regions, except this is on the short arm, and then this is just showing the relative locations of everything on the long arm of the chromosome. Now, like I said, this is kind of leading us into rearrangement of chromosomes and leading into the next chapter, which is gene expression. So histones, like I said, remember those are the proteins that the DNA is kind of wrapped around and creates those nucleosomes, and then this allows them to be tightly compacted. Well, they can undergo chemical changes. Okay, so they can undergo chemical changes where a functional group is either added or taken off, and this is called acetylation. It's an acetyl group, or deacetylation. So this results in the reorganization of chromatin. So it kind of reorganizes it so that there are regions which will be more tightly packed versus other regions. When you want it loose or less tightly packed, this is where you want to start to express the genes that are in that region. So dense packing of the heterochromatin makes it very difficult for the cell to express, right, the genes that are located in those regions. And what I mean by express, and that's why it's in quotation, is that means using it to create its product. Okay, this is called gene expression. Okay, so gene expression means to express the gene or basically produce the product that it codes for. And it's usually a protein, but again, like I said, it could be an RNA. type of RNA. So gene expression involves two processes. Okay, and this is leading us into chapter 17, which I'm not going to get into today, but this kind of introduces it. Okay, so chapter 17 discusses the whole process of gene expression, and it involves two major steps. It's called transcription and translation. Transcription is basically the use of the DNA in the chromosome to make RNA, make a certain type of RNA. The RNA that's made from that is going to be used to make protein, if it's meant to make protein. Okay, so in order to perform transcription, it needs to be not tightly packed in that region. So if there's a gene that needs to be expressed, let's say the cell needs to make a particular protein, that area of the gene will be modified, right? Okay, the chromatin will be reorganized or modified so that it will be loosely packed so that it can be used to make the RNA, which will eventually make the protein. Okay, so the next chapter, which will be next week, we will talk about chapter 17, and that is gene expression. So we're going to talk about the details of the process of transcription and the details of translation. We're going to talk about mutations in proteins and how they affect, or mutations in DNA and how they affect the proteins, the product of the proteins. All right. So today, okay, if you remember, we talked about this is discoveries that led to all this information. Okay, so the discoveries about DNA, and we learned about a bunch of different scientists. So some of these include that DNA is the genetic material, or it's the transforming material. It's transmitted from one generation to the next, and it stores the information which directs the processes that occur inside the cell. DNA, again, is transforming material, so it's able to be transmitted from one cell to the next, and that's where it was seen in the R and S forms. So the heat-killed S form, the DNA was basically incorporated into the live R form, which is the non-pathogenic, actually transforming it into something that's pathogenic. There's composition differences between species in terms of different bases, the percent of adenines, thymines, cytosines, and guanines. And that there's a relationship between adenines and thymines. So there's an equal percentage or composition of adenine and guanine, or adenine and thymine, and guanine and cytosine. And remember, these last two are related to Chagav's rules. And then the molecular structure, Watson and Crick and Rosalind Franklin and Wilkins led to that. And then, of course, the molecular model, or the structure of DNA, the whole DNA molecule, the double helix, and how it's connected, explained Chargaff's rules, what he found. And the semi-conservative model of replication. Okay, so we learned how it was discovered, how DNA is actually replicated. And then, of course, I talked about the replication process and all the proteins that are involved in that. And then, of course, the differences between eukaryotic cells and prokaryotic cells. And we talked about the function of telomeres and differences in how chromatin is structured differently in different regions of the chromosomes. And what's not mentioned here is the repair machinery, proofreading and repair. So I did talk about that as well. So yes, it was a lot of information. I would recommend, if you haven't read this already, go ahead and read about this, complete that table assignment. I'm going to make sure that it's actually posted in the bonus module. And then what I would do is definitely, before you attend next Monday's discussion, read chapter 17. It's another loaded chapter with a lot of information. So if you've read it, or at least perused through it before, it'll make a lot more sense. It'll be easier to understand. So are there any questions about DNA replication? And I know I need to revisit the differences between leading and lagging strands, so I will do that. But I don't have anything else new for today.